So is this on? Everybody can hear me? Thank you so much for that introduction. It's a pleasure to be here today. I will be speaking about work from my research team that focuses on the aging of immune cells and the consequences of intervening on functional pathways within those immune cells. So this audience is well associated with the idea of low-grade chronic inflammation that accumulates during aging, where the big picture idea would be the increase in chronic stimuli that accumulates with aging, activating and driving chronic long-term inflammation within cells like macrophages, B cells, and T cells, forming what we refer to as inflammaging. This drives the actual cellular pathology and the symptoms that we see in older organisms, where the aging of the immune system is referred to as immunosenescence. And all of this feeds into a dysfunctional immune response, including reduced health span and increased morbidity. And of course, a few years ago, the COVID-19 pandemic, where older individuals had 100-fold greater risk for death, really highlighted the need for understanding the aged immune system and understanding more specifically which pathways we can target to improve that response. If we look at the various immune cell subsets, we can describe specific changes within B cells, within T cells, or within myeloid cells. I'm not going to go through all of these right now, but I want you to understand that we really can identify a decline in the function of all of these cells within older individuals, and this is referred to as immunosenescence. Major signs of immunosenescence include the involution of the thymus, an increase in memory lymphocytes, increased low-grade chronic inflammation, specifically activation of the NLRP3 inflammasome. And we know that senescent cells are a major source of that inflammation, as seen by an increase in senescence-associated secretory profile. It's not really entirely clear what proportion of immune cells are senescent or not, but I know this is a major hot topic in the field. So I wanted to introduce you to the model that we use, which is referred to as a dirty mouse model or normal microbial exposure model. And this was developed at the University of Minnesota by Steve Jamison and Dave Mousequist, along with Sarah Hamilton. And the concept is taking the specific pathogen-free mice that have a limited exposure to any pathogen and co-housing or transferring the fomite from a pet store mouse to that SPF mouse. And in this manner, we generate mice with a diverse microbiome and a diverse immunological repertoire. So young mice handle this new exposure without any difficulty. There's nearly 100% survival. The immune cells become activated, and there's a development of memory in the young. However, what we found when we challenged 24-month-old SPF mice to this enemy exposure, there was 100% lethality by day 14. And so we use this as a model to really examine the resilience of the immune system. I'm not going to go through the extensive work that went into this huge collaboration between the University of Minnesota and Mayo Clinic, but I wanted to show you two key experiments, where the first is an experiment where we identified the virus that is present in the transfer from the pet store mice, and this is called MHV, which is a beta coronavirus in the same family as SARS-CoV-2. And we either left mice unimmunized or immunized them with an MHV. So in black, you can see that the mice that are left unimmunized have 100% lethality, where in blue, if we give these mice MHV immunization, there's complete protection from that lethality, indicating that exposing or generating awareness in the immune system of which virus is present permits the mounting of an appropriate adaptive immune response. We also performed experiments to target senescent cells using senolytics. And here I'm showing you the use of besetin, which is going to deplete out senescent cells, where you can see that treatment with besetin improved the survival of those old mice. We also did these experiments in transgenic mice and using the senolytics dasatinib and quercetin. So we did additional experiments to show that this increased survival is mediated through a reduced cytokine storm, including reduction in SASP. and improved adaptive immune function, so increased antibody production. And this generates a model where in young individuals, they're able to mount an appropriate response to the virus, including an antiviral response, where the immune cells can generate an innate and adaptive response, producing antibodies and mediating survival, where this dysfunctional response leads to an accumulation of senescent cells, an increased cytokine storm that really impairs the ability of the innate and adaptive immune response. So depletion of the senescent cells or immunization reduces that cytokine storm and permits the development of this adaptive immune response. And you heard from Dr. Kirkland yesterday about the number of senolytics that are currently in clinical trial, and that really supports the movement of these therapeutics into human patients. The next question we wanted to ask is which immune cell contribute to the dysfunctional response? We did a lot of work to really characterize the immune cells using flow cytometry and sequencing. So what I wanted to focus on was this finding that exhausted PD-1 positive CD8 T cells are increased first in aging and then further amplified with exposure to NME. So CD8 T cells are normally a cytotoxic T cell that are responsible for clearing viral infection. And you can see we found this amplification in the liver, but also in the spleen, the lung, and the mesenteric lymph node. And we found that other subsets of T cells also showed increased exhaustion of PD-1, suggesting that those show amplified exhaustion. So what exactly is exhaustion? We know that these cells have increased profile of inflammatory markers, as seen here by an increase in granzyme K and CCL5, but they have reduced expression of granzyme B that allows them to mediate their cytotoxic capacity. In our experiments, we measured granzyme B, and using two different models of NME, we found that the old mice had reduced ability to produce granzyme B, which was consistent with a reduced functionality of these exhausted cells. But it actually gets more complicated than that, where exhausted cells can be described as a whole subset of heterogeneous T cells, and we can use markers like CXCR5 or KLRG1 to subset out these exhausted cells. So this is a Tisney analysis that shows that we found comparable levels of heterogeneity between the young and the old. And this was actually exciting to us because it implied that even in old individuals, we could use checkpoint blockade or different therapeutics to target those old exhausted cells. And that's sort of further emphasized by this quantification of CXCR5 here, where you see that there's comparable frequency of CXCR5 positive cells in the young and the old. So I told you this is a good thing, right? But there's actually a problem with activating these immune cells. If we increase their function, we're increasing their cytotoxic function, but we're also potentially increasing their inflammatory function. And so it's a double-edged sword where we could have heightened inflammation and exacerbated tissue damage and adverse effects. So in our model here, we have an old mouse where the increased exhaustion is preventing the clearance of those virally infected cells. And if we use a checkpoint blockade or an anti-PD-1 treatment, one outcome is that we might see increased tissue damage and increased inflammation because of the increased T cell activity. But we might also be able to improve T cell function and improve their cytotoxic capacity, increasing their ability to remove infected cells. So we asked this question performing a survival experiment where, going back to our model of NME exposure, old mice were either given an isotype control or the anti-PD-1 to block PD-1 signaling on the CD8 T cells. And we found that this anti-PD-1 treatment increased survival as compared to the old PD-1 treated mice. We also were able to use a model of intervention where instead of giving PD-1 prior to the NME exposure, we gave it after the NME exposure, where we saw a consistent increase in survival. And how is this actually mediated? We measured granzyme B in the young and the old male and female mice. So prior to exposure to NME, granzyme B is quite low. And after exposure, you can see that young mice have an increase in cytotoxic capacity. The old mice show a reduced cytotoxic capacity and reduced granzyme B expression, but anti-PD-1 treatment increases granzyme B across both sexes. So the model that we have from this story is that in younger mice, we have survival, we have a reduced frequency of PD-1 positive cells and increased granzyme B, and reduced inflammation. We did experiments where we were able to show that anti-PD-1 had no effect in young mice, and we think that this is because there's actually no presence of PD-1 positive cells. In contrast, in old mice, we see mortality and exposure to the enemy, where this is mediated by the increase in the amplification of the PD-1 positive cells, a reduction in granzyme B, and an increase in inflammation. So what's important here is that checkpoint blockade increased survival, and it did this by increasing the cytotoxic capacity of those CD8 T cells. We know we did experiments and we showed that it required the CD8 T cells and not the CD4 T cell subset, and that granzyme B is mediating this protection. This is obviously really interesting and I think relevant to the field, where there's a number of high-impact papers that have revealed the potential of PD-1, PD-L1 pathway for improving immune function and reducing the senescent cell. And so we think that targeting the immune cells in combination with senolytics or senotherapeutics could be a realistic avenue for older individuals. There's a little bit of work out there on trying to address this question, where there's some research and meta-reviews that have looked at the efficacy of checkpoint blockade in older individuals with cancer. And overall, they show that there's comparable efficacy of checkpoint blockade in older individuals, for those 65 years of age or older, as compared to the young. And to me, this supports the idea that checkpoint blockade could be useful in the context of just normal aging, or in the context of acute infection, where we could eliminate senescent cells and improve the function of those CD8 T cells. I wanted to, I'm not going to spend a lot of time on this, but I wanted to point out a recent paper from our lab where we have expanded on this idea of how B cells are impacting the response to endotoxemia or sepsis. And in these experiments, we asked how B cells could mediate that change using lifelong deficiency of B cells, so MUMT mice. And we found that lifelong deficiency of the B cells had no impact on lifespan, in parallel to work that was produced from the Weiner lab. But when we challenged these mice, there was a reduction in cytokine production and a reduction in disease severity as measured by body temperature. And so we think this supports our ideas of the immune system as being a complex, interactive system, where we really need to understand the pathways within each immune cell and how it could be contributing to different aspects of endotoxemia or sepsis. I also wanted to highlight a poster that will be presented on Thursday as a sort of upcoming project from my lab, where we are looking at the GDF3 SMAD axis in macrophages during endotoxemia and sepsis. So this work is based on work that I generated as a postdoc in Vishwadeep Dixit's lab, where we identified GDF3 as a top-regulated gene by age and the NLRP3 inflammasome. So this highlights the idea that some genes are increased in combination with an increase in inflammation, and that the reduction in chronic inflammation also reduces those genes. So GDF3 is a cytokine in the TGF-beta family. We know that it regulates lipid metabolism through ALK7 signaling. We also did experiments to show that it regulates the NLRP3 inflammasome activation through signal 1. But how and whether it's mediating age-related inflammation or age-related inflammation in the context of endotoxemia and sepsis is something that we've taken on as a project. So we think there's a lot of potential avenues for understanding the impact of GDF3, where previous work over the last several years have shown that GDF3 is linked to multiple diseases, including Alzheimer's disease, myocardial infarct, and NASH in the liver, and obesity. But it's quite complicated. As you can see, some of them promote the disease, and some GDF3 promotes some of these diseases or worsens some of these diseases. And we think this is based on the inherent functions of GDF3. I'm not going to go through the data, but I wanted to give you the overview where we think that this is a gene expressed in macrophages, and when macrophages become chronically inflamed, GDF3 is elevated. And we have shown that GDF3 can regulate the SMAD2/3 pathway, and using mouse models where we genetically manipulate GDF3 or pharmacological inhibitors where we can block SMAD3 signaling or block BRD4, we have revealed a role for GDF3 activating the SMAD signaling and regulating cytokine production, the development of these inflammatory macrophages, accumulation of B cells within various tissues, and then disease severity in old mice. We've also done experiments where we can show what GDF3 is not involved in. So for example, proliferation and recruitment of those macrophages. And just to sort of bring it all home, I hope I've convinced you that the immune system is complex, but this complexity is exciting because it gives us multiple ways to target it. The changes in the immune system with age can actually give us new avenues to target because old immune cells are inherently different than the young immune cells. So our goals are really to improve immune cell function, increase immune resilience, and overall, in the long goal, improve tissue function. And I will just leave my acknowledgement here and thank you all for your attention.
Thank you so much, Christina. So we have time for one or two questions. Go ahead.
I have a question. So I remember there was a paper that came out in, I think, JAMA Dermatology or something like that, showing that with PD-L1 inhibition, or I can't remember if it was PD-1 or PD-L1, that people got their hair color back. I'm not sure. Have you seen hair coloration come back in these mice, or graying of hair impacted?
Yeah, that's really interesting. Right now, the experiments, the way we've done them, wouldn't be set up to address that because it's been quite an acute treatment of the anti-PD-1, but we are very interested in the long-term effects because I think even if acutely there's tolerance of anti-PD-1, the long-term effects are also important to evaluate.